Trials that involve group randomization or delivery of interventions to groups are very common in public health and medicine. This is particularly true for behavioral interventions. Very common to see group randomized trials or cluster randomized trials, which involve group randomization, or individually randomized group treatment trials, which involve delivery of interventions to groups. So we're going to talk about all of those today. We'll start by talking about randomized designs for clinical trials. There are 4 that we're going to cover, and then we'll turn to analytic methods. We'll talk about sample size and power, and we'll spend some time talking about alternative designs that are often considered in lieu of randomized designs. At the end of this section of the course, you should be able to distinguish among the designs that are commonly used in NIH-supported clinical trials. You should be able to choose among those designs based on the methods that you are planning for assigning participants to study arms and delivering your intervention to participants. Those methods are extremely important for dictating the type of design that you should use. You should be able to identify the analytic methods that are appropriate for the design that you choose. And while I don't expect everybody to become a methods expert and do their own analysis and sample size, you should at least know the questions to ask and be able to recruit collaborators who have those skills to work with you to implement your trial. So the first section that we'll cover is the design of these studies. There are 3 main classes of designs that I'm going to talk about. Randomized clinical trials, individually randomized group treatment trials, and group randomized trials. For the last one, there are 2 subcategories, parallel group randomized trials and step-wedge. So there are really 4 designs that we'll talk about. In an individually randomized control trial, and this is the one that you're all familiar with, this is the one that you've studied already, individuals are randomized to control or intervention arms. More complicated designs, there may be more arms, but this is the basic setup. So here we have a group of individuals. We have a randomization process to place them into intervention and control. And observations are assumed to be independent from the participants before and after randomization. We can assure this if the participants don't interact with one another, if they don't interact with a shared intervention agent, if they're really just completely separate from one another after randomization and once the study begins. There is a variation on this design, which we call individually randomized group treatment trials. We start in the same way. Individuals are randomized to either control or intervention arms. So here's the same pool of participants. We have a randomization scheme. They land in the intervention and control arms. So far, it looks just like an RCT. The difference is, at this point, participants in at least the intervention arm will receive at least some of their intervention in a group, often a small group, or from a shared intervention agent, here depicted as a healthcare provider. So the 3 on the left are interacting with a particular healthcare provider. The 3 on the right are interacting with a different healthcare provider as part of their intervention. In this particular slide, the control arm doesn't have any of that sort of interaction with healthcare provider. So this might be a waitlist control or a no treatment control. Treatments are delivered in groups or via shared intervention agents. That is the critical defining component of the IRGT, individually randomized group treatment trial. The last general category is the group randomized trial. This is also called cluster randomized trial. So I tend to use group randomized trial. It is exactly the same thing as a cluster randomized trial. You can use those terms interchangeably. The distinguishing characteristic for the group randomized trial is that we're working with pre-existing groups usually. Sometimes we create the groups just for the study, but more often they're pre-existing. And those groups are what are randomized to either the control or intervention arms. So we have the groups, we have a randomization process, those groups land in the intervention and control arms. And our expectation is that the observations from participants in the same group are correlated before randomization, during the study, after randomization. Correlated observations is the expectation in a group or cluster randomized trial. As I said, there are two kinds of group randomized trials. In the parallel group randomized trial, we have separate but parallel intervention and control conditions through the duration of the trial, and there is no crossover from one arm to the other. So participants stay either in the intervention arm or they stay in the control arm. The alternative group randomized trial is a stepped wedge group randomized trial. All groups start in the control condition. They cross over to the intervention condition on a staggered schedule in a random order. And by the end of the study, all groups are in the intervention. So that is what separates parallel and step wedge group randomized trials, and we're going to talk about both of these. Let's start by talking about the risk of confounding. In the RCT, the randomized control trial, there's usually very good opportunity for randomization to work. We usually have a large number of participants that we're randomizing. Most RCTs have more than 100. Many have thousands. And if you have a large trial and the randomization occurs correctly, confounding is not usually a concern. Potential confounders are evenly distributed among the study conditions by virtue of the randomization. In the individually randomized group treatment trial, there may be less opportunity for randomization to work because these are often smaller studies. If they're just as large, not a concern, but if they're smaller, confounding could be more of a concern. In the parallel group randomized trial, confounding is the rule, not the exception. These studies often have a limited number of groups that we're randomizing, either groups or clusters. And if you have 20 groups or clusters that you're randomizing, or 30 or 40, chances are pretty good that once you complete your randomization, there are going to be some differences between the two study conditions, the two study arms. In any single realization, there is limited opportunity for randomization to work. And that is particularly true if the number of groups that you're randomizing is less than 50. In the step wedge group randomized trial, instead of having our groups nested within study arms, they're actually crossed with study arms. Everybody begins in the control condition and gradually steps over to the intervention condition so that all the groups and all the participants provide data under both conditions. That means that groups are crossed. And as soon as we do that, we avoid most of the confounding that would be of concern in a parallel group randomized trial. At the same time, the intervention effects are substantially confounded with calendar time. And that's because all of the groups are in the control condition at the beginning. Gradually, over time, more and more groups move to intervention. And by the end of the study, all of the groups are in the intervention arm. So the intervention itself is confounded with calendar time. That means we've got to adjust for calendar time in any analysis in a step wedge group randomized trial. I would argue that the step wedge design is inherently less rigorous than the parallel design. And so I tend to discourage people from using it. Sometimes a parallel design is not appropriate. And in that case, you can certainly consider the stepped wedge. But if you can use the parallel design, it will usually be faster and less subject to the risks that we face in the step wedge design, and therefore preferred. Most parallel group randomized trials and individually randomized group treatment trials involve just one treatment factor. So I call it study condition. A lot of people call it study arm. Most studies have just two levels, intervention and control. You could have more. Most cross condition with time so that we're following either the same participants or the same groups over time. And the pre-test, post-test design is the most common variation. Most parallel group randomized trials employ a cohort design, though some use a cross-sectional design. And I've done studies where we had both a cohort and a cross-section in the same study. Almost all individually randomized group treatment trials employ a cohort design. I'm really hard-pressed to think of one that used a cross-sectional design. And individually randomized group treatment trials vary in two important features, their nesting structure and their membership structure. And we're going to talk about those details shortly. So nesting structures in an individually randomized group treatment trial. There are three nesting structures that we see. The one in the middle, the partially nested structure, is the most common. The open squares are the control participants, the closed squares are the intervention participants, and the gray circles are the intervention agents. In the partially nested design, we have intervention agents working only in one arm. That would be the intervention arm. And they interact with participants in that arm. There are no intervention agents in the other arm, so that intervention agents are nested in a single arm. That's why it's called a partially nested design. If we look to the left, we see the fully nested version. Here we have agents in both arms, different agents in the two arms, and therefore it's a fully nested design because we have different agents in each arm. On the far right, we have the third possibility, which is a cross design. Now the agents are working in both arms, not just in one arm, so they're not nested at all. The agents are crossed with arm, and agents interact with participants in both arms. As we will see as we go along, there are substantial advantages if you can use a cross design, particularly in terms of sample size and power, and also in terms of analysis. But there are risks because since the same agents are delivering both interventions, you have to make sure that they deliver the correct intervention to each participant. So contamination is a bigger concern with a cross design. We're going to talk about three membership structures. The first is the single membership structure. That is characterized by having each participant interacting with just one intervention agent. In the partially nested design shown in the middle, there's a single line coming from each participant, the square going up to the circle, the agent. So each participant is interacting with a single agent. But we have more than one agent in that arm. On the left, in the fully nested situation, again, the participants interact with just one agent. We have agents in both arms, multiple agents in both arms. In the cross design, each participant interacts with a single agent, but the agents are working with participants in both arms. We can also have a multiple membership structure. This is simply a more complicated version of what we just looked at. The difference is that each participant interacts with more than one intervention agent. So if you look at the squares and the lines going up to the circles, each square will have, in this case, two lines going up to two different circles, indicating that each participant interacts with two agents. It could be far more complicated than this. And this is considered an example of a multiple membership structure. The last structure that I'll show you is one that we see but don't recommend. Here there is a single agent, either in the only arm that has agents, or in each arm, or a single agent for the whole trial. In the partially nested situation, there's a single agent responsible for delivering the intervention to all of the participants in that arm. The reason that we don't recommend this design is that that completely confounds the agent with the intervention, and you can't distinguish the effect of the agent from the effect of the intervention. So it's not something that we recommend, but we do see it, and so I'm showing it to you. On the left, we have the fully nested case where there's a single agent in the intervention arm, a different single agent in the control arm, and on the right, we have the crossed case with a single agent for the whole study interacting with all of the participants. Again, we don't recommend the single membership structure, single agent structure. Let's talk a little bit about types of step wedge designs. The step wedge was introduced in 1987 in the Gambia hepatitis study. The first methods paper by Hussey and Hughes appeared in 2007, so not all that long ago. And this design has become very popular over the last 10 or 15 years. We see a cross-sectional version of the design as well as a cohort version. In the cross-section, different individuals are measured at each data collection point. In the cohort, we have the same individuals. Many of the papers distinguish between closed and open cohorts. So I identify that there. And a good paper is the Hussey and Hughes 2007 article. The basic step wedge design is shown here. The randomization of groups is to sequences. So in this case, we've randomized 2 groups to each of 4 sequences. The groups are numbered in order, but that doesn't mean that we took the first 2 and put them in sequence 1 and the second 2 and put them in sequence 3. We randomly assign the groups to the sequences, and then for convenience, we just label them 1 through 8. We have a number of periods in the design. In this example, there are 5. In the first period, as is standard in step wedge designs, we're collecting data from participants in each group, but nobody's getting any intervention yet. Beginning with period 2, the groups assigned to sequence 1 begin the intervention. In period 3, the intervention continues in sequence 1 and begins in sequence 2, and so forth. So you have this sort of stepped entry into the intervention condition by the groups based on the sequence that they were randomized to. And by the time we get to period 5, all of the groups are in the intervention. You can see that each group provides data under both intervention and control. And that means that if it's a cohort, each participant provides data under intervention and control. If it's cross-section, they're only providing 1 data point, and it may be intervention, it may be control. Time is completely is quite confounded with the intervention in step wedge designs. This is always true, and it's because of the staggered implementation. Nobody's in the intervention at the beginning, everybody's in the intervention at the end. It's essential that you adjust for time in any analysis of a step wedge design. There is increased risk of within-group contamination in a step wedge because the groups transition from control to intervention. Some groups may want to start earlier than they're supposed to, so that can be a risk. Some groups may not get started right on time when they're supposed to. That can be a problem. Important to monitor for evidence of contamination to allow you to do an as-treated analysis if you see it. How do you choose among the designs that I've offered? We've talked about 4, the RCT, the parallel group randomized trial, the IRGT, and the step wedge group randomized trial. The first question to consider, is there a strong rationale for randomizing groups rather than individuals to study conditions? And if the answer is no, then we consider a second question. Do participants receive their treatment in a group format or from a shared intervention agent? And if the answer is no, you've got an RCT. Congratulations, it's the easiest design to implement, probably the least expensive and smallest, and carries the fewest problems. So I always recommend using an RCT design if that is possible. If, however, participants receive even just some of their treatment in a group format or from a shared intervention agent, then you've got an IRGT trial, and you need to deal with the issues that IRGT trials face. We've talked about some of those on the design side. We'll talk about more of them on the analysis side. If there's a strong rationale for randomizing groups rather than individuals, and the usual rationale is you're worried about contamination. The reason that we do school-based studies, randomizing schools, is that we know that the students in the school are going to interact with one another. And if we try randomizing individuals, participants in the intervention arm are likely to talk to participants in the control arm over the course of the day, or certainly over the course of the year, the school year. So to avoid contamination, we randomize whole groups or clusters, in that case, schools. So if there is a strong rationale for randomizing groups, usually worries about contamination, then we can consider the second question on the right side. Is there a strong rationale for rolling out the intervention to all the groups before the end of the study? I would argue usually not, but sometimes there is a good reason for doing it. If there is not, you've got a parallel group randomized trial. If there is a strong rationale, then you've got the step wedge design. So these are the 3 questions to consider as you're trying to choose among the 4 designs that we've discussed. And all of this is presented in a paper in the Annual Review of Public Health published in 2020. A couple of other points I want to make on the design side. Um, a priori matching and or stratification are good strategies to use. I recommend them quite generally. They can be used in any of the four designs that we've discussed. The use of you can use either one to ensure balance on potential sources of bias, potential confounders. If you think that the intervention effect is going to differ, let's say among men and women, among urban and rural, um, then you want to stratify on those factors so that you have a sufficient number of participants or groups or clusters in each of the strata so that you can look at differences between the strata. A priori matching is useful if the matching factors are well correlated with the endpoint. But the choice often will depend on the number of groups available and the expected correlation. If you've got a small study, less than 100 participants, for example, stratification is generally recommended. And in most of the studies that I do, I try to use stratification and avoid matching. Another strategy that you can use is constrained randomization. Stratification and matching are difficult to use if you've got a bunch of things that you want to control for in the randomization process, and constrained randomization is a good solution for that. You look at all possible allocations of groups to arms based on the scoring that you get on the variables that are included in the constrained randomization. You identify the allocations that provide good balance, and then you choose one of those allocations at random from that subset, and that's the allocation scheme that you use for the trial. Fan Lee published a couple of papers, 2016-17, that describe this process and show that constrained randomization can improve power, maintains the type 1 error rate in parallel group randomized trials. So this is a strategy that I recommend. In the parallel group randomized trial, it's before randomization. In the to arms, in the step wedge design, it's before randomization to sequences. That's the end of our discussion of design issues, and the next topic that we're going to take up is analytic issues.